Welcome. I'm Will Conrad, Assistant Professor of Chemistry and Molecular Biology at Lake Forest College. I'm reporting to you live from the Molecular Biology Resource Center here at Lake Forest. Actually, it's my kitchen table, Illinois Cylinder Shelter-in-Place orders. Nonetheless, I'm here to answer 3 key questions about the CDC's coronavirus tests.
Specifically, I'm here to answer why testing is so important, what kind of test should be used, and how the RT-PCR test currently being used works. Testing is the best way right now to limit the spread of coronavirus. Without controls in place, each person infected with the novel coronavirus infects an average of around 2 people. If unchecked, this results in exponential growth in case numbers. 1 case leads to 3 cases, 3 cases leads to 7 cases, 7 cases leads to 15 cases, 15 cases leads to 31 cases, and on it goes with exponentially more people getting infected in each round. By testing a case and all their contacts, those contacts with the disease can quarantine and prevent transmitting the disease further. This is called contact tracing. Contact tracing worked in South Korea. Notice the South Koreans rapidly increased testing in orange even when there was only a handful of cases in blue. South Korea continually ramped up testing to a rate of over 10,000 tests per day to identify every coronavirus positive contact and reverse the spread of the virus. So testing, especially early on, can contain the virus and stop its spread. Testing saves lives.
So we know that testing can be really important for slowing, stopping, or reversing the course of the disease. But what test should we use?
The test needs to be highly sensitive. It needs to detect the virus in the subject's body before they become infectious. It also needs to be highly specific, not detecting common related viruses that cause minor symptoms. The way to detect the virus earliest is to detect the virus itself, so we need to target something the virus is made of, specifically lipids, proteins, or RNA. Coronaviruses are made of a protein and lipid coat. The lipid is depicted here as a black line. The spike protein is in pink, and the coat protein is in brown. Inside are other proteins in blue, and single-stranded RNA in red, which serves as the genetic material used to hijack cells to make more virus. In order to directly detect the virus, you need to detect one of those materials. The viral lipids are collected directly from their human hosts, so it would be nearly impossible to use those lipids to specifically test for the presence of the virus. How would you know if you were detecting virus or human lipids? Also, there aren't very sensitive tests for lipids. Proteins can be readily detected by specific antibodies. Antibodies are frequently used in diagnostic tests and can detect as little as 10 picograms of protein. However, my back-of-the-envelope calculation suggests that 10 picograms of protein would require 301,000 viral particles. When you add into that the fact that it takes a lot of R&D time to ensure the antibody is specific to its protein but not to other related viruses, and to scale up the manufacture of that antibody, it looks like a less appealing option. RNA, on the third hand, can be detected with high sensitivity and specificity. The RT-PCR test, designed by CDC, can detect as few as 1 viral particles per microliter and can be made highly specific. I used a government online search tool to confirm the primers designed for detecting coronavirus are not found in humans. Furthermore, the chance that a sequence perfectly matches one of the 2 primer sets due to chance is 1 in 8.7 times 10 to the 40th power. Not very likely this probe will match a different random piece of RNA. This is important if you're only collecting a few viruses isolated from a nasopharyngeal swab.
An infected patient will carry virus in the back of the throat, so the nasopharyngeal swab is the most reliable way to collect virus and its RNA from a possible coronavirus patient. It's easy enough to say we should detect RNA, but how do we actually detect it? And what part should we detect? Let's take a look at the complete viral genome and see what the CDC designed.
The complete sequence of the novel human coronavirus was published in 2019 by Wu and colleagues. Here I've depicted the viral single-stranded RNA as a pink line of 29,303 nucleotides in length. Brown arrows below this pink strand depict polypeptides encoded by this sequence, aka proteins. The RNA binding protein depicted in the coronavirus is highlighted in blue. The spike protein is in pink, and the envelope is in orange. The three red dashes in the bottom right corner are the locations of the probe CDC designed to detect the virus. Because of how RT-PCR works, it can be easiest to detect sequences at the far end of an mRNA. Currently, they are only using probe sets N1 and N2, the outside two dashes. Let's zoom in on this map to the sequence level to see how probe 1 was designed and how it works. Here I've drawn a 106 nucleotide region about 1,000 bases from the end of the viral genome. The A, C, U, and G represent the bases adenine, cytosine, uracil, and guanine present in RNA, along with a ruler for scale. This region of the viral genome encodes for a protein called nucleocapsid phosphoprotein, which is a key structural protein that holds the viral RNA inside the viral envelope. The single amino acid sequence for this protein and a ruler is translated below the RNA sequence. It makes sense to design a probe against a critical viral protein because mutants in this region are less likely to produce viable virus. Thus, a probe is more likely to stand the test of time as the virus mutates. Finally, depicted here is the 72 nucleotide region that is specifically detected by CDC's primer set 1 in red. This primer set contains a forward primer that writes DNA from left to right in a PCR reaction, and a reverse primer that writes DNA from right to left as depicted here. Remember, both of these primers write in a 5' to 3' direction from their respective templates. In between the two primers is a probe that is written in the same orientation as the forward primer. This probe contains a fluorescent molecule called 6-carboxyfluorescein, or FAM for short, and a quencher molecule called black hole quencher. Black hole quencher stops FAM from glowing green when it's nearby. However, whenever the forward primer is elongated, polymerase separates FAM from BHQ, and FAM is able to glow green.
Now that we've looked at the anatomy of CDC's probe set, let's look at how it works through RT-PCR to detect the presence of coronavirus RNA.
First, RT-PCR uses DNA, and as I mentioned before, coronavirus is an RNA virus, meaning it uses RNA for its genetic code instead of DNA. To convert RNA to DNA, we use a viral enzyme called reverse transcriptase. Reverse transcriptase makes a complementary strand of DNA to the RNA, which can be amplified by RT-PCR. The cDNA is complementary to the viral RNA. In coronavirus, the viral RNA is akin to human messenger RNA. It's called positive strand RNA because it carries the coding sequence that can be directly translated into protein. Once cDNA is generated, polymerase chain reaction begins. PCR is traditionally a 3-step cycle between high-temperature denaturing, low-temperature primer annealing, and medium-temperature extension. Let's look at that 3-step cycle, denaturing, annealing, and extension, in detail. First, the sample is heated to near boiling, 94 degrees Celsius, to make all DNA single-stranded. After the denaturing step, samples are briefly cooled to an annealing temperature to allow the primers to specifically bind. Because the cDNA is complementary to the messenger RNA, both the forward primer and the TaqMan probe specifically anneal to the cDNA. The primer provides a site for the enzyme DNA polymerase to read the cDNA and write a new strand of DNA. DNA polymerase detects the 3' end of the forward primer only because the 3' end of the probe is blocked by black hole quencher. When polymerase extends the DNA sequence from the forward primer, its exonuclease activity frees the FAM fluorophore into the surrounding solution. In traditional PCR reactions, this polymerization occurs at the optimal temperature of Taq polymerase, 72 degrees Celsius. The CDC protocol runs at the annealing temperature of 55 degrees Celsius, which is common for RT-PCR reactions where only a short sequence needs to be written by Taq polymerase. It shortens the length of the entire assay. After extension, the cycle begins again with denaturing at 94 Celsius. Now denaturing is separating two strands. In this second round of PCR, during the 55 degrees Celsius annealing step, the forward primer and probe bind again to the cDNA. Now the reverse primer also has a complementary sequence in the strand of DNA synthesized in round 1. After the primers anneal to the cDNA and the new strand, the extension round begins. DNA polymerase extends from the 3' end of the forward primer and from the reverse primer. Again, polymerase releases a FAM molecule from the forward-facing probe. The number of free FAM molecules doubles from 1 to 2. Notice how the amplification off the reverse strand does not release any fluorescence, but will become important in the third and in all subsequent rounds of PCR. Round 3 begins again with a heat denaturation step. Now there are 4 strands of DNA. In the second step of round 3, the sample is cooled to 55 degrees Celsius for primer annealing. Forward primers and the probe bind the 2- DNA strands, which are written right to left, 5' to 3'. Reverse primers bind to the positive strands, written left to right in the 5' to 3' direction. In the extension step, polymerase binds to the 3' end of all 4 annealed primers and writes DNA in a 5' to 3' direction. Because 2 strands are compatible for binding the forward primer and probe, 2 FAM molecules are released in this round of PCR. With each round of PCR, the number of free fluorescent FAM molecules doubles. In round 1, it was 1 molecule total. In round 2, it was 2. In round 3, it was 4. This fluorescence doubles with each cycle. This is exponential growth of signal. After 30 rounds of PCR, a single viral RNA molecule is duplicated into over 500 million copies and 500 million FAM fluorescent molecules. The RT and RT-PCR stands for real time because the total fluorescence of the reaction is measured with each cycle. This real-time measurement can be used to quantify the original amount of DNA template. The high sensitivity of RT-PCR is due to this exponential amplification of signal. The CDC PCR protocol takes about 45 minutes to complete 45 cycles. The overall process takes several hours, including RNA isolation from the nasopharyngeal swab. Abbott Labs has modified this PCR protocol to occur at 1 temperature using different enzymes, and can detect coronavirus directly from the swab in less than 5 minutes.
So, RT-PCR can turn 1 copy of viral RNA into hundreds of millions of copies of DNA. But how do we turn that signal into a proper test?
It's not enough to simply see the result from one probe set to provide a diagnosis. A properly controlled experiment must be performed to make sure the probe amplification is specific for coronavirus. Every complete coronavirus test consists of 2 different PCR reactions to test for coronavirus at 2 different locations, probe sets N1 and N2 from before. It also contains a positive control PCR probe set to test for the presence of human RNA that should also have been collected from the nasopharyngeal swab. 12 samples can be run side by side for coronavirus probes 1 and 2 and the human RNA probe. Each run contains a no RNA negative control, 10 separate patient samples, and a positive control that contains a known quantity of viral RNA. This is an example of a qPCR run of such a setup. Let's interpret the results. The x-axis depicts PCR cycle number, and the y-axis depicts a measure of fluorescence. Remember, we expect fluorescence to increase exponentially with each cycle number if template DNA is present for the probe. Let's imagine that in this experiment, patient samples 1 through 9 were negative for coronavirus, and patient sample 10 was positive for coronavirus. What results would we expect for a properly run experiment? First, we would expect no amplification in the negative control samples, the samples where no RNA was added. If we see amplification in the no RNA samples, we can conclude that the samples were contaminated and all results will be deemed inconclusive. We also observe no amplification for coronavirus negative patients number 1 through 9.
We observe amplification in all patient samples for probe set RP, which tests for human RNA. If we do not observe amplification of human RNA, we can conclude that there was an error in collecting RNA from the nasopharyngeal swab, or that the RNA degraded and would have to collect a new swab from the patient. We also observe amplification in the condition where we add probe sets 1 and 2 along with known viral RNA. This proves that our RT-PCR reaction works and that all of the enzymes and probes are active and sensitive. Finally, we observe amplification of probe N1 and probe N2 from RNA collected from patient 10. Because we know that there was no amplification in the no RNA condition, we can conclude that this signal does not come from contamination. Importantly, we can strongly conclude that patients 1 through 9 are negative for coronavirus. Because we observed amplification of probe N1 and N2 using known viral RNA, we know the coronavirus probes are working properly. Because we observed amplification of human RNA, we can know that the patient samples 1 through 9 are negative and not just lacking RNA. Using these appropriate controls, we can conclude that the patients 1 through 9 are negative and patient 10 is positive. Thanks so much for watching this video. I hope you learned more about why we test for coronavirus, why we test for RNA, and how that test works. If you'd like to learn more about molecular biology at Lake Forest College, please click the link shown next. If you're able, please contribute to support teaching and research at Lake Forest College. A special thanks to the RCSB Protein Data Bank for their depictions of coronavirus, reverse transcriptase, and Taq DNA polymerase. I adapted them for this presentation. References will also follow this presentation.